aspects of okay, I'll start again. So welcome everyone for another session of the Brain on Wednesdays, where we look at the different aspects of neuroscience as regards the various psychiatric illnesses. And today we have a very interesting topic going to be presented by Dr. Sagar Bhalke, and the topic is the first session on brain bioenergetics. And to share this session, we have Dr. Shobha Nair. Dr. Shobha Nair has been my colleague and friend for God knows how many decades now. And she is at present the head of psychiatry at BARC Hospital, Mumbai. She has clinical experience of more than 25 years, and she's a PG teacher for DNB and currently the vice president of the Bombay Psychiatric Society. A brief introduction about Dr. Sagar Bhalke also. He passed his MD from Nair Hospital, and he's currently senior resident at the Department of Psychiatry, Sion Hospital, Mumbai, and his interests include molecular psychiatry, genetics, and everything neuroscience. I would hand over the mic to Dr. Shobha to say a few words about the topic, and then when she signals, we can start the session. Over to you, Shobha.
Thank you so much, Dr. Malay. Nice to be sharing this platform with you. Biochemistry was a very tough subject for me in MBBS 1, and I'm sure many of us had the same difficulty as compared to anat and physio. And it was this persuasive president, Ruksheda, who has forced me into chairing this session today. When I took up psychiatry, I thought I am done with biochemistry, only to understand eventually that I better learn, and all of us need to learn. To understand biology, we need to also learn biochemistry. So today's fascinating topic on brain bioenergetics, which is biochemistry of energy metabolism in the brain. And I thought I would ask some questions, which could be what our speaker, Dr. Sagar, would tell us in the next two sessions. One, is this energy metabolism different in the brain as compared to other organs? If so, how is it different? Why do we need to understand and study it? What is the current hypothesis of the various neurodevelopmental and the neurodegenerative diseases that we see in clinical practice? I mean, all of us learned in younger classes in school that, what is a powerhouse of the cell? And then we answered mitochondria. And the currency of this chemical energy being ATP. So whether dysfunctions in these pathways, what does it lead to? Can we sense this dysfunction? Can we measure it? Are there markers? And whether this understanding, how close are we to translate this understanding into understanding of disorders and their management in clinical practice. So over to you, Dr. Sagar. We are all eagerly waiting to learn more as to how our brain cells function. Thank you.
Sir, may I start?
Yeah, so before you start, everybody please keep yourselves on mute for the entire duration of the presentation. And there's a chat box, so whatever questions you have, you can put it up in the chat box and we'll take them once the presentation is done. Be sure to write your name when you ask a question, because sometimes it's only iPad and Samsung and things like that, so we don't have people with those names in India at least. So over to you, Dr. Sagar. The stage is yours.
Thank you, ma'am. Thank you, sir. I'll be sharing my screen now. Malay sir, I'm not able to press a minus on it. Is this okay?
You can get it
Slide
to
it
one
off.
side. That's it. Done.
Okay. So, welcome everybody to this introductory session on brain bioenergetics. As ma'am had already told all of us, this session will mostly contain some principles and facts about biochemistry. So, the pillars of this session will be physiology and biochemistry. So, starting with the basics, what does energetics mean? Although the term sounds quite fancy, bioenergetics, it is just a study of energy transfer between different tissues, uh, transduction of energy between the different tissues. And as we all are psychiatrists, neurologists, or somebody else who is a clinician, our primary target here is brain, and hence we'll be focusing more on the bioenergetics of the brain. Uh, Before we move ahead with specifically brain bioenergetics, let us first understand what does cell rejuvenation mean. So the fundamental unit of body is a cell, and it is the aim of all the clinicians to enhance the functions of cell. And whenever there is an impairment, we have to correct it so that the cellular function gets back to normal. So there are three steps. First is systemic way in which we can modulate cell rejuvenation by reducing inflammation, improving the nutrition, or modulating the blood factors. Then we can modulate the cellular function and subcellular mechanisms. For today's session, our target will be subcellular rejuvenative methods, and out of which primarily mitochondria, which has been shown to have effects on metabolic manipulation, cellular reprogramming, and ablation of senescent cells, that is apoptosis. So we'll move ahead with the primary topic, that is brain bioenergetics. Let us first of all start by knowing some basic things about what the brain is, how much ATP it requires in proportion to the energy that our body requires. So brain consists of 2% of the body weight, but it consumes 20% of energy even at rest. That is, even if a person is sleeping and if a person is consuming 100 ATP, 20% of ATP are consumed by the brain. Out of those 20% which are used by the brain, 15% are used for general cell function. That is, whether the cell is from liver, from kidney, from heart, or brain. All the cells have some general function like ionic stability, maintenance of cytoskeleton, calcium homeostasis, for which only 15% is used. Rest of the other thing, energy is used for specific brain functions. Out of the 20% that the brain consumes, 70% of energy is required by neurons, and the rest of the energy for glial cells, especially the astrocytes. Maximum expenditure of energy in the brain happens at synapses. So, at the place of synapse, mostly depolarization of the action potential happens. And after that, we need to restore the membrane potential. And that is where the maximum energy is spent in the brain. Brain controls whole body energy homeostasis. Now, what does this mean? Apart from controlling its own ATP consumption, brain also controls how much ATP is consumed by the rest of the body by regulating the intake of food, by regulating our motor activity and exercise. Metabolic heterogeneity of mitochondria is something that we'll be seeing in the further slides. The next point is brain function is highly efficiently coded. So what does this mean? It means that for any specific cognitive task that a brain does, brain is designed in such a way that the task is done with as minimal number of neurons as possible. So the brain is highly efficiently coded for energy consumption. Despite that, it has been found that cognitive tasks increase the energy consumption by just 100 to 20%. So I'll just give a simple example. For example, if a person is at a resting state and he is consuming 100 ATP. Now, what the studies are saying is that if the person is now engaging in a cognitively higher task, the person is only consuming 110 to 120 ATP, which means that cognitive tasks are responsible for just 10 to 20 ATP consumption. So, who is consuming this 100 ATP at the basal stage? So, it is nothing but the resting activity and basal neurotransmission, which means that even if a person is not performing any active so-called cognitive task, still maximum amount of energy is used in that resting activity. So we must have seen the EEG strips on which we usually look for the spikes on the EEG. But background activity ठीक है थोड़ा सा कुछ है and we ignore that. But what is this background resting activity? What is this basal neurotransmission which is consuming a lot of energy? Initially, people used to say that this is just white noise. This is just something that does not have any function. But gradually, we have come to know that this high resting activity, even at the absence of cognitive tasks, actually is involved in emotional processing, memory consolidation, reprocessing of past events, pre-processing of the future events, assessing the environment, and expectations of the reward. And we know that emotions, memory, reward networks are involved in various mental health disorders. So there is a higher role of resting activity and basal neurotransmission as far as mental health disorders are concerned. So let us see where all this energy is spent inside the brain. There are two specific sections. First is to restore the things, and second is for maintenance. So what are the things that need to be restored and which require energy? Ions, neurotransmitters, second messengers, vesicles, and molecules. And some maintenance work also requires energy, such as cytoskeletal maintenance and glial cell function. Now I will ask you to look at this figure on the right and uh, would like to know what your observations are regarding this, if anybody is up for it. For your information, the green one is a nucleus and the fluorescent purple ones are mitochondria. So any observations that you people have? Anything that anybody wants to comment regarding the appearance of mitochondria or their behavior? Sir, shall I go ahead?
If you look at the chat box, two people have mentioned, Dr. Vaibhavi mentions that some protein synthesis might be going on. Dr. Nachiketa mentions that dendrites and synapses consume more energy. So maybe those are the processes related to synaptic transmission that is going on. So you can
Yes.
carry on from there.
Yes, yes. So I was not able to see the chat box. My apologies. So yes, those are the correct answers. But apart from that, some very simple observations that we can make out from this video are that a cell does not contain just one mitochondria. Contrary to the diagrams that we used to draw in the school, कि एक nucleus है, एक Golgi body है, एक mitochondria है. So a single cell on an average has around 500 to 5,000 mitochondria inside it as compared to a single nucleus. Second thing that we can see is the movement of, the movement of mitochondria is much faster as compared to the movement of the nucleus, which means that the basal activity in mitochondria is more as compared to nucleus, translating into the fact that mitochondria, although are the powerhouse, they themselves also require a lot of ATP to function themselves. Another thing that we can see here is mitochondria are changing their shape. So we have been always taught that mitochondria are bean-shaped, cylindrical shaped, but that is not the fact. The shape of mitochondria varies according to their function, according to their location, and the stage of the development. So they are changing their shape. Next thing that we can see is most of the mitochondria are attached to each other. So mitochondria in the average cylindrical shape are usually attached to each other to form a mitochondrial network. And that is how mitochondria communicate with each other. Sometimes there is fusion of mitochondria whenever there is need, and sometimes there is fission of the mitochondria also. One more thing that we can see in this diagram is most of the mitochondria is situated near the nucleus. That is because mitochondria need to regularly communicate with the nucleus. Uh, this communication is essential so that components of oxidative phosphorylation are formed properly. Some parts of oxidative phosphorylation are formed by mitochondria and some by the nucleus. So a good communication between them is essential, and that is why most of the mitochondria are always located nearer to the nucleus as compared to nearer to the cell membrane. So I'll go ahead. Some basic details, discovery in 18th, 19th century, but during 1950s, 1960s, people came to know that mitochondria is involved in beta oxidation, Krebs cycle, oxidative phosphorylation. But now in the current century, people are finding it out that sirf powerhouse ही नहीं है, but there are different non-energetic roles that the mitochondria performs. And we will be looking at those functions in the further slides. So this is one more illustration of mitochondria on the left, where we can see the typical bean-shaped mitochondria. Let us now talk about the phylogenetics of mitochondria in brief. There are various organelles in our cells which are derived from other organisms like Golgi bodies, mitochondria, and some part of our nucleus, which is derived from viruses and bacteria. In the similar vein, it has been now established very well that mitochondria are derived from alpha proteobacteria. So around 200 to 50 million years ago, the eukaryotic cells engulfed some alpha proteobacteria. There was inadequate degeneration of the proteobacteria. So some part of the proteobacteria remained inside the eukaryotic cell. So what happened is the genetic material of that prokaryotic proteobacteria, it entered the nucleus of the eukaryotic cell, and there the genetic material of both the cells got integrated and the function got remodeled. Some genetic material of proteobacteria did not enter the nucleus. वो nucleus के बाहर ही रह गया, and it formed its own membrane, which gradually evolved into the cell organelle that we now called as mitochondria. On the right side is the diagram, again showing green colored mitochondria and red colored lysosomes. So I have just added these videos because we need to know that the things that we are studying are seeable, observable, and we will be trying to formulate interventions or at least formulating interventional targets on things which are real. So again, we can see in these two diagrams, multiple mitochondria per cell, lot of activity, cylindrical shape, some shapes changing, mitochondria forming networks, most of them nearer to the nucleus, which brings us to the concept of mitochondrial heterogeneity. So mitochondria differ in terms of their shape, in terms of their composition, in terms of their function, and in terms of their energy production and energy usage. Along with that, mitochondria also have shape and variation of shape and size according to the day. So there is diurnal variation in the mitochondrial appearance also, which is dependent upon the amount of light falling on it. So we'll be seeing about that in further slides. So the famous diagram, Mitochondria has different parts, three parts that we must know. First is the outer membrane, which is more permeable to small molecules, proteins, and ions. Then the inner membrane, which is very tightly bound and not permeable to almost everything. The cristae, which are the site for oxidative phosphorylation, and the matrix where Krebs cycle happens. So coming to the important part of this session, the Krebs cycle. During initial days of my UG, I thought that Krebs cycle is like a mountain that I have to climb, so that I need to pass the exam. I need to memorize all the names of the enzymes and the steps. But I have come to realize that Krebs cycle is nothing but a series of 8 to 9 enzyme catalyzed reactions, which is designed to break hydrocarbons to produce CO2, water, and ATP. So we can see in this diagram here that water, CO2, and red colored ATPs. So there is a break of hydrocarbon leading to formation of CO2, water, and ATP substrates. Now, one interesting thing about tricarboxylic acid cycle is its other name is citric acid cycle. The reason why citric acid was chosen in the evolution was because it has 3 carboxylic acid components. So this carboxylic acid is specifically chosen by evolution because it has high energy storage capacity. Secondly, it is stable in the human physiological conditions. And thirdly, because it is broken up very easily. And lastly, because it acts as a precursor for all the other compounds present in the Krebs cycle. So that's the reason why citric acid is the primary substrate through which Krebs cycle occurs. Now somebody might say ki, if citric acid is so important a substrate for Krebs cycle, does it mean that if we consume a lot of lemon, yeah, citric acid, will it enhance the mitochondrial function? So I don't know the answer currently. Next thing we need to know about is electron transport chain. So electron transport chain is a chain present in the inner mitochondrial membrane where there are two things which are transferred. First is transfer of electrons from donor to recipient. And second transfer is of proton molecules outside, eventually leading to formation of the ATP. So this is the function of electron transport chain. So why is it important to know how electron transport chain works? Because when electrons are transferred throughout this chain, sometimes the electrons are leaked outside. If a few electrons are leaked through the ATC chain, these are cleared up by peroxisomes, sometimes lysosomes. But if the cells are working over time, there is an increased leakage of electrons through this electron transport chain. And the peroxisomes are not able to handle these electrons or the reactive oxygen species. And then the inflammation in the cell starts, which can ultimately lead to symptoms of anxiety and depression. So to see if there is a medical student who is very afraid of exams and exam pass में आ गई है और 7 दिन से रात को सो नहीं रहा है और सिर्फ पढ़ाई कर रहा है, which means that the student is studying daytime also, nighttime also. So the brain is working in an overdrive, not getting enough rest. So if the brain is working more, it translates into neurons working more. Neurons working more translates into mitochondria working more, and mitochondria working more translates into electron transport chain working more. So ETC chain is working more, so electrons get leaked at a greater amount, and those are not cleared off by the peroxisomes and leading to further inflammation and precipitation of various abnormalities like depression or anxiety. Second thing that we also need to know about electron transport chain is it is composed of complexes 1, 2, 3, 4. And these complexes are also helped by vitamin K, coenzyme Q, which brings us to the fact that good nutrition is quite important. Sometimes due to nutritional deficiency of some vitamins, magnesium, or coenzyme Q, the electron transport chain does not work properly, and there is inadequate ATP generation, leading to symptoms of lethargy, mitochondrial hypofunction. Let us talk about the energy then. Uh, Glycolysis, everybody knows, happens in cytosol. Two ATPs are created. Krebs cycle with ETC chain oxidative phosphorylation leads to formation of 32 to 34 moles of ATP. As compared to cytosol, where only two ATPs are generated per glucose, in mitochondria, 32 to 34 ATP are generated. So this is one thing where mitochondria succeeded in enhancing the cellular function in terms of bioenergetics. Before mitochondria was present in eukaryotic cells, the energy production was too less and higher functions were not possible. Now, just some trivia. I have added this slide from a paper. Everybody can go through it later. I'll share you the slides. Basically, what this slide mentions is that uh, the amount of ATP produced by mitochondria is in trillions. And hence, mitochondria is rightfully termed as the powerhouse of a cell. So, what are the sources of energy for mitochondria? Glucose, fatty acid, amino acid. The uses are transport of ions, transport of neurotransmitters and other chemicals, heat generation, and ATP synthesis. So, regarding formation of mitochondria, we are studying about the Krebs cycle, ETC chain, formation of mitochondria, because eventually we will be seeing that any impairment in any of these steps can lead to mental health disorder in the long run. So, biogenesis. So, what does that mean? Increase in the size of the mitochondria and the number of the mitochondria, which is controlled by various transcription factors, hormones, steroids. Traditionally, it was thought that mitochondria was just the powerhouse of cell and its only function was generation of ATP. But then a finding came about in 1970s, 1980s that mitochondria release cytochrome C. And cytochrome C was found to activate the DAMP and caspases pathways. And caspases are involved in the apoptosis. So whenever there is a lot of inflammation and stress in a cell, mitochondria release cytochrome C, leading to apoptosis. Therein the revelation came about कि mitochondria has multiple functions apart from energy dynamics. We also need to know about signaling pathways because there are three reasons. First is that our medicines, that is psychiatry medicines, we know work by secondary and tertiary signaling pathways. For example, PKC inhibitors like endoxifen or lithium. So we need to know about the signaling pathways of mitochondria also. In the similar vein, let us now understand about hypoxia. So whenever the cell has hypoxia, there is formation of reactive oxygen species. And in reactive oxygen species generation, there is hypoxic gene expression. So whenever the cell is in hypoxia, the genes are expressed, especially the hypoxia-inducing factors. So what these factors do, they try to reduce the activity of mitochondria as much as possible so that further damage is not done. In the sense that they inhibit the fission, they alter the ETC components and make them hypoactive. There is a metabolic shift. So oxidative phosphorylation, say it gets converted into glycolysis. There is upregulation of antioxidants. So herein lies the importance of using antioxidants in the inflammatory brain disorders. So what is normal mitochondrial function then? So it depends upon who is studying mitochondria. Some will call it as powerhouse. Some will comment about its shape, about its network activity. Some will say that it is involved in apoptosis. Some will say that it is just an endosymbiont bacteria living inside a human cell. So these are various mitochondrial functions. I will just zoom it a little bit so that everybody can see. So mitochondria contribute into the function of nucleus on the left, and on the right we can see that it acts as a substrate producer for neurotransmitters, hormones, RNA, DNA, lipids, and proteins. And along with that, it also helps in other systems too. This is an exhaustive list of functions that mitochondria does, and we will only see the headings of it so that we understand the scope of this topic called bioenergetics. So, membrane potential generation, amino acid metabolism, ascorbate metabolism, bicarbonate metabolism, calcium uptake and extrusion, hydrogen sulfide detoxification, heat production, intermediate metabolism, light focusing. So it has been shown that mitochondria are receptive and sensitive to light too. Lipid synthesis, sodium import, neurotransmitter synthesis and degeneration, oxidative phosphorylation, oxygen sensing, permeability transition, protein import, respiration, reactive oxygen species production, steroid genesis, antiviral signaling, apoptotic signaling, crystal remodeling, DNA signaling, interorganal communication, mitochondria to mitochondria communication, mitochondrial dynamics, motility, and vesicle formation. So the point that I'm trying to drive home is that mitochondria does a lot of work apart from ATP generation, and we need to understand all these functions and then try to formulate any interventional targets accordingly. Let us now study about the genetics of mitochondria. So mitochondria has a circular mini chromosome, which we can see on the right side. It contains 37 DNA genes, encoding for 13 polypeptides, which form the ETC chain or the oxidative phosphorylation. As compared to the human genome, just a minute. Malay sir, am I audible?
You are
Yes,
audible.
you are
Yeah,
audible.
yeah, no problem.
Yes. So, as we can see on this slide, mitochondrial DNA contains only 37 genes. Compared to that, we can see that human genome contains around 20 to 25 genes, 25,000 genes. So the amount of genes in mitochondria is quite less. Also in the diagram, we can see that the base pairs in mitochondria is only 16,000. Compared to that, the human nucleus genome contains 3.2 to 3.3 billion base pairs. So compared to the genetic material in the nucleus, the mitochondria has a very small amount of DNA content. So if that is the case, why is it important to study the genetics of mitochondria if the genetic content is so less? That is because in human nuclear DNA, there is a larger component of so-called junk or non-coding DNA. But in the mitochondrial DNA, the non-coding part is only 7%. The rest 93% is coding, which means that 93% of mitochondrial DNA has some significant function, especially during the oxidative phosphorylation. So the margin of error is very less. Any small mutation in mitochondrial DNA can lead to mitochondrial disorders. There are 250 known mutations in human mitochondrial DNA. Compared to that, in the nucleus, if there is mutation, the homeostasis and the buffer system of body tries to take care of it, but very less so during the mutation in the mitochondrial DNA. The diagram below can show some so-called antidepressants and their effect on different ETC chain components, which some drugs increase the activity, some decrease the activity, and some have no observable effect on the mitochondrial ETC chain. Secondly, we saw in the table of the functions of mitochondria that there is mito to mito communication and mito to nucleus communication. So we can see here that the nucleus is communicating with the mitochondria for formation of oxidative phosphorylation and TCA cycle. And the byproducts of mitochondria like ROS, heat, and ATP are giving feedback signaling to the nucleus. So this is a bidirectional relationship between nucleus and mitochondria. So let us go ahead with the impairments in bioenergetics. We saw how mitochondria are formed, the genetics of mitochondria, its functions, a Krebs cycle, and also ETC chain. So at any of these places, if there is an impairment, it leads to cellular malfunction, which can lead to brain dysfunction and further behavioral abnormalities. So there can be impairment in nuclear DNA, which is the major site of mitochondrial protein synthesis. There can be impairment in cytoskeletal transit, that is the connection between nucleus and the mitochondria. There can be direct mutation in the mitochondrial DNA, in Krebs cycle, in ETC chain, or electron supply. So one or more of the impairments in these chains can lead to impaired bioenergetics. Moving ahead to the last few slides of the talk today, uh, involvement of mitochondria in aging. So we can see in this diagram that if there is a mitochondrial dysfunction, it can lead to dysregulation of nutritional sensing, loss of proteostasis, epigenetic alterations, genomic stability is impaired, which can eventually lead to stem cell exhaustion and early aging. Exercise has been shown to boost mitochondrial function by release of exerkines in the blood. So there is improved bioenergetics, improved mitochondrial content, improved mitochondrial dynamics, leading to neurogenesis, neuroplasticity, and improved cognition. Nutrition has a huge role in the mitochondrial function. First is basic nutrition, carbohydrates, fats, proteins, vitamins, all are needed so that the Krebs cycle and ETC work properly. So uh, the next is alcohol use disorder. So whenever a person consumes alcohol in a heavy pattern, the brain energetics shift from glucose to acetate. Gradually, the person starts eating less food and consume more alcohol, and acetate acts as the substrate for the mitochondria. And this acetate has been shown to cause the symptoms of withdrawal when the patient does not consume alcohol. So some people have now started suggesting that ketogenic diet is useful for management of withdrawal, but the evidence is inconclusive at this moment. Calcium also causes dose and time dependent inhibition of the mitochondrial bioenergetics. Uh, this session was mostly designed in a way that we understand how the mitochondria works, and there won't be larger focus on mental health disorders as of now, but still in the coming few slides we will see what the role of mitochondria and bioenergetics is in mental health disorders. It will be just like a glimpse of the upcoming sessions. So there is impaired basal neurotransmission activity. So we saw in the earlier slides that basal neurotransmission activity consumes more than 80% of the ATP in neurons. And it is important in emotional processing, memory consolidation, re and pre-processing of past and future experiences, prediction of environmental states and rewards. So all these are impaired in schizophrenia, depression, bipolar disorders. Hence, impairment in basal neurotransmission is something that can be an interventional target for us. In depression, it has been seen that there is impaired ATP production rate. There is reduced mitochondrial enzyme levels, especially cytochrome C reductase, succinate cytochrome C reductase. There is reduced glucose utilization. And imaging studies have also shown that there is reduced ATP production in depression. There is elevated reactive oxygen species, increased mitochondrial superoxide levels. As we saw that there is an increased reactive oxygen species, and peroxisomes are not able to clean those up, leading to increased inflammation, leading to depression. In rat depression models, it has been seen that imipramine elevated the mitochondrial numbers. So what they did is they injected rats with injections of lipopolysaccharides, and it led to depression in rats. Imipramine diya and they got better. In some models, they injected IV mitochondria itself, and it also reduced the depressive symptoms in the rats. There is a role of energetics in chronic fatigue syndrome. In CFS, all the measures of energy capacities are weaker, lower energy rates, and lower mitochondrial outputs. In psychosis, there is reduced ATP production in the depressive phase, and there is increased ATP production in the manic phase. There is reduction in N-acetic acid, which is the viability marker. On the right side, we can see the diagram of ATP synthase. So why we are studying this is because it has some relation with lithium. So the way ATP synthase works is the hydrogen atoms go inside this rotor, and then eventually an ATP is formed. Lithium is known to reduce the ATP synthase activity, which is present in the mitochondrial matrix. So if lithium is inhibiting this ATP synthase, it leads to reduction in the formation of ATP. And we saw that in mania, ATP are formed in a larger number. So lithium helps in reducing the ATP formation and a resolution of the manic features. So this is one of the mechanism through which lithium works. Then chromosome 22 has a lot of mitochondria localized genes. So any small or large mutation in that can lead to impaired oxidative phosphorylation and eventually reduction in the ATP production. This has been linked to depressive phase of bipolar and negative symptoms of the psychosis. Coming to the last but one slide. So a little bit about cancer too. In cancers, the patient has adequate availability of oxygen and the mitochondrial machinery is intact. But despite that, in cancer, there is a metabolic shift from oxidative phosphorylation to glycolysis with ROS accumulation and anti-apoptotic signaling. So the cells do not die. So mitochondria are not able to release the cytochrome C properly, and eventually the cancer proliferates. Last slide of today's session. So what are the things that we need to learn and we may talk about in the further sessions is bioenergetics of other systems. This is important because brain's bioenergetics is very closely related to bioenergetics of liver, of kidney, heart, adipose system, and skeletal muscle. We also know that heart is the second brain, or liver is the second brain, kidney is the second brain. So understanding the bioenergetics of other organs is equally important. We will also see some proxy markers through which we can measure the mitochondrial activity. We will talk about sensitivity of mitochondria to light, to hormones, because we already know that hormones are important in the synthesis of mitochondria. We'll also see sensitivity of mitochondria to non-psychiatry medication, role of energetics in mental health disorders, and current and future interventions in bioenergetics. Okay, with this, I end my session. There are three major sources through which I prepared for this presentation. First is this book on the left, which is Clinical Bioenergetics by Dr. Sergei Jostojic. Very comprehensive. I'll share it with all of you. Second is if somebody is more eager to learn, then there is a separate journal called Frontiers in Neuroenergetics, which has a lot of papers, a lot of recent advances where they are releasing new things about brain bioenergetics. And if somebody has less time but still is curious about brain bioenergetics, I would suggest that you go to Twitter and just use these keywords and you will come to know a lot about neuroenergetics. So here I end my presentation. Thank you.
Congratulations, Dr. Rahul, for bringing to us a lot after studying a lot of stuff in books and in research papers, concising it and getting it on this platform and helping us understand more about how the mitochondria functions and what is its important role in brain diseases. I think the points that you highlighted, if I may just conclude, is that you've told us how a lot of energy is required not only for the functioning of the brain, but how the brain itself controls the whole body energy homeostasis, and how 20% of the energy is utilized by the brain, though it is a very small organ. And you have also told us that when it comes to mitochondria, unlike what we studied, there is a lot of heterogeneity, that it changes its shape, its functions, and the metabolism also can keep changing, and that you may be having different genetic predispositions, and each person may have a different set of functioning and structural mitochondria. You also emphasized that for baseline functioning of the brain, a lot of energy is required, both at rest as well as when awake or during sleep, and that additional cognitive tasks demand very less energy. I mean, we didn't understand why it is so, because we all thought that when we are engaged in a lot of cognitive tasks requiring rigorous cognitive activity, we do tend to get fatigued. Fatigue sets in, right? So we feel that there is a lack of energy and we need to rejuvenate. But apparently research says that it's the baseline activity itself which requires 70% of the energy consumption, and only slight increments in energy is needed to do cognitive tasks. So that you emphasized with, and also you said that how mitochondria doesn't function just on its own, but there is a lot of interaction with the nucleus, and how the mitochondria evolved. I mean, evolutionarily, how it evolved because of some bacterium, how the eukaryotype eventually led to the present mitochondria that we have, and how it can produce multiple times the type of the energy levels which was not available to our evolutionarily
Ancestors. Yeah.
older ancestors. So you emphasized on the Krebs cycle as well as the electron transport system, electron transport chain, and you have actually conveyed to us about depression and other psychiatric disorders more as inflammation. Means how inflammatory pathways eventually leading to apoptosis or cell death can be what we understand by through psychiatric disorders, as well as degenerative disorders, or what age can do, how aging can impact. So what you want to say is that everything eventually is at the level of mitochondria, right? And in the next presentation, we would learn more about how drugs work on it, and how other energy systems of other organs are distinct from that of the brain, and learn more about what are the future horizons in probably developing molecules which target this, and also understanding disorders of the brain. Thank you so much.
Thank you.
So may I just look whether there are any questions? Well, a lot of people have appreciated your presentation. Any questions?
So one question. There are two, I think.
Yeah, in the process of neuroplasticity. Apart from handling oxidative stress, what is the role of mitochondria in plasticity? Neural plasticity or adaptation?
Yes. So one thing that we saw during the presentation was mitochondria has continuous communication with the nucleus, that is the DNA from the nucleus. And the genetic content from mitochondria and genetic content from the nucleus both have the function of protein production. So any change in the mitochondrial genetic content or nuclear content or both can lead to altered protein production. And basically altered protein production translates into changing the networks and connectivity in the brain, which is called as neuroplasticity.
Okay.
So what did you what was the take-home message from that neuroplasticity?
So, sir, the take-home message would be that neuroplasticity can be contributed to either by changing the nuclear genetic content or through changing the mitochondrial genetic content, or both. And evidence has now started creeping in that our so-called antidepressants and some antipsychotics have started tweaking this genetic content, and that is how they bring about the change, that is neuroplasticity.
Okay, so if I can add to that, so whenever there is any neural process, whether it is anything, whether it is a synaptic process or whether it is whatever is happening in the individual neuron or the glial cell. Okay, now what happens at the mitochondrial level is few things. One is if you want to maintain a plastic change has to be maintained. It's not a passive process. If you leave it to passivity, there will be something called as long-term depression, means that plastic change will go back. So one is to maintain the dynamic nature of the plastic change. So you will require a different kind of different level of energy available, more or differently than what the original cell has, because now it has to maintain that connection. So what is going to happen is this, that one is at the mitochondrial level, all the processes that will go into making that plastic change stable at a new level everything will be more. That is one. Second thing, for more energy to be available, you need more mitochondria, or you need the existing mitochondria to work more. So there will be changes in mitochondrial morphology and physiology. So one of that is, as you mentioned before, there is going to be change in the, first of all, where they are localized. So if you are going to maintain something near in a synapse, you will have more number of mitochondria there. Second thing is, with this dynamic change, the shape will change, the networks will change. Okay. Then the connectivity with the nucleus will also be changed, because to maintain that will require two things. One is things which are going on, and you will also need some signaling so that the DNA makes more different kinds of proteins or more proteins, so that that thing remains still over a period of time. Because probably there is nothing long time sustainable if there is no corresponding change at the DNA level. Now, for that to happen, even the mitochondria around the nucleus will undergo the same kind of change: number, morphology, connectivity. That is one thing. Second thing is that mitochondria do not function alone. You mentioned about the connectivity with the nucleus, but it also it works in a network with lysosomes also. So whenever we have this excess something in the cell, okay, in terms of, because when something is overworking, the wear and tear is more, right? Any wear and tear is more vesicles are formed, more vesicles will break down, more proteins are working, more proteins will break down. So they all need to be thrown out of circulation where lysosomes will come in. Even when mitochondria are working more, many of them get damaged more because wear and tear is more. So they work very closely with lysosomes. They form those lysosome mitochondrial complexes. So that will be, kind of taken care of. So it's not only energy production is more, even the scavenging function is more. Now, when all these things are happening, it has to maintain the redox potential. Because if that redox is not maintained, okay, the cell will collapse. The mitochondria will collapse first, then the cell will collapse. So to maintain that redox potential, okay, so a lot of things will happen, because you need to maintain that new kind of normal, that is the plastic change. So this is what is to be understood from mitochondrial function. Yeah, some more questions have come up, Dr. Shobha.
Yeah. I mean, is there translation into actual practice? Somebody has asked whether, what is the nutrition for mitochondria? I mean, is there anything that we can actually ingest which will help improve the health of mitochondria?
So I personally don't know any particular form of nutrition which will directly enter into mitochondria.
Correct.
But as we saw in one of the slides, basic nutrition which consists of carbohydrates, vitamins, fats, lipids is essential
So
for
what is good for the whole body is good for mitochondria.
because
What is
body is
good
made
for
up of
us is good for
cells.
mitochondria. Absolutely.
Yeah.
Then there's another question whether we can say that the decreased energy and fatigue in depression is due to a mitochondrial dysfunction in energy production.
Yes. So
Can we
many
conclude
studies
that,
have come
or
out.
it is just a hypothesis?
So, ma'am, many studies have come out now which have shown that there is a reduced number of mitochondria per cell in the patients of depression, and some studies have also shown that the even the normal amount of mitochondria which are present have are uh, functioning at a suboptimal level, leading to lethargy and other symptoms of depression.
So is it a cause or is it a consequence?
It is a cause,
It is one of the cause.
one of the yeah.
Hmm.
See, again, coming to that, in this basic understanding of mitochondria today, maybe you would cover it next time when you talk of the illnesses in particular, is that, when we talk of this multiple genes involved in the associated with the psychiatric illnesses, many of these relate to mitochondria. So it's one of the basis for mitochondria not functioning well is the genes which are shared,
Polygenic.
which come in the thing, because there are many proteins involved. There are many proteins which are involved in mitochondrial physiology, or in whatever the processes mitochondria are doing. Okay, and even separate mitochondrial genetics. So as you mentioned, mitochondrial genetics is two. So one is the nucleus of the cell, which has got certain mitochondrial related genes, and the second is the mitochondrial DNA itself. Now both these have different origins and both of them function differently. The genes in the DNA of the cell, which are responsible for mitochondria, these genes give the structure of the mitochondria. So in outer membrane, inner membrane, mitochondrial proteins. Okay. So the DNA of the cell contributes to this structure. But the energy production, the electron transport chain, is exclusively derived from the mitochondrial DNA, which are those small circular DNAs which are floating in the matrix. And each of this circulating DNA, each of these floating DNAs is different. So there is a lot of permutation and combination even within a single cell. And no two cells will have a similar distribution of mitochondrial DNA. So one thing is fixed, which is there in the nucleus of the cell, and this mitochondrial DNA, which is variable. So what happens is that in the nucleus, if something is not okay, it can have a problem on mitochondrial structure, which may not be much. But if there is something problematic in the mitochondrial DNA, then the effects are magnified, because you mentioned that there are only a certain number of proteins which are very important vis-a-vis the function of the mitochondria. So small change in mitochondrial DNA, large and amplified change, and this amplification leads to a lot of problems which are not only related to brain, but then they involve entire systems. So the way of looking at pathology is that brain bioenergetics comes at the top, other organ bioenergetics comes at a slightly lower level, because the main consumption of energy is by the brain. So small changes in mitochondrial DNA, problematic changes. Okay, let's say that little problematic change happens when there is a problem in maintaining the redox. Now, small change, because brain utilizes so much energy, neurons utilize so much energy, that small change in that is going to have a cascading like effect on the functioning of the neurons. But in other organ systems where the energy utilization is less, that little change may not be clinically significant. That's why when it comes to the production of symptoms, okay, we have got, if there is some mitochondrial involvement, we get primarily psychiatric symptoms. If there is lots, we get brain involvement with other systems involvement. So it is also about the shared genomic issues between disorders, the mitochondrial DNA. Second thing is how mitochondria, in terms of inflammation, how it handles itself. One of the things that happens with mitochondria is you mentioned about energetics. So all the substrates go into the cristae, and then energy ATP is generated. Then some electrons leak, and then the peroxisomes and
Peroxisomes.
the dismutases, they take over and they scavenge it. And if it is more, then it can lead to oxidative issues and oxidative damage and things like that. Now, with aging, one, and with a high level of inflammation that sets in, this process is going to reverse. So more energy that was now being available for reactions and things like that is now spent in repairing the mitochondria. So available energy, what the mitochondria should do, is less. The problem is in the mitochondria itself. That's why the energy suffers. So the initial thing is that the reactions will go into reverse. So more energy will be diverted towards internal repair processes of the mitochondria. If the mitochondria are fused and they're working fine, now they will get separated. So fission will occur. If the fission is still causing the mitochondria, I mean, it's not good enough, then the mitochondria themselves undergo death. It's mitochondrial lysis. So this is how the mitochondria work. So at all levels, whether it is nuclear DNA, mitochondrial DNA, at energetics, maintaining the redox potential, the inflammatory status, whenever there is inflammatory status, the mitochondria shift the metabolism from
Glycolysis.
glycolysis to the kynurenine pathway. And this kynurenine pathway is pro-inflammatory. So it again kind of multiplies the inflammation. So there are a lot of things when it comes to depression or mania. The mania might have more ATP production, but because of that more ATP production, there is more of inflammatory change, the less of more of oxidative damage, more of everything else. But the sum total is what happens with depression also. So pathways, even in the mitochondria, are different whenever there is mitochondrial involvement, and so many things. So I think you will cover all these things. But this is what we need to understand. It's not simple one thing which is only bioenergetics, because people will get easily fooled into saying, okay, more mitochondria, less mitochondria. Then we will have things like, okay, make mitochondria healthy. Mitochondria will not get healthy if the mitochondrial DNA is damaged or it's an issue. So when it's a question of nutrition, the mitochondria should be in a position to utilize that nutrition. If primarily there is some fault in their physiology, then it will not work. So I think initially somebody had asked a question.
Cause and effect.
Not cause and effect. With Shobha's permission, I'll ask you, what improves mitochondrial nutrition and what is the mitochondrial cocktail?
I will leave this question to Malay sir and Bhaskar sir only to answer.
So no, when you talk about what targets are, then it will make sense.
No, at this moment, I should not say mitochondrial cocktail comes. After next
Yes.
presentation, we should discuss about
Only when we have the targets identified and what are the strategies, then you can think of how it works. Because again, it's not a universal formula.
Right.
It will vary from person to person, and even in the same disorder from various people, from person to person. So that's not the thing. Then yes, Shobha, you can take the other questions.
There's just one question which I think he has already answered, whether energy is utilized only by cells or also for transport between cells, intercellular, long distance. I think you clarified that,
So
right?
Nachiket Bhai had asked it. So energy is not just utilized by the cells, but as we had discussed, most of the energy is actually spent
the
at
synapse,
the
no?
synapse.
Yeah.
So especially for after depolarization to maintain the action potential again to the resting state.
Yeah, I think that's all as far as the questions go.
Okay.
Overall, everybody has liked your presentation, Dr. Sagar. Participants learned. So, brain health is akin to mitochondrial health.
Yeah. So
learn more next time.
Yes, because this is one way of integrating what we know in terms of symptoms, in terms of processes, whatever processes we know of, little or more. In simple words, we start with neurotransmission and synapse, and then we go on to what are the second messengers, and then what happens at the DNA, and how plastic changes happen, and how degeneration happens. One of the unifying processes are the mitochondrial processes. Because anything that requires energy, what are the things that require most energy? And I'm not talking of systems. What are the most important things that require energy and huge amounts is one, the nucleus itself. Because it is in the charge of so much
Everything almost.
everything. It has to produce proteins of all kinds. It has to guide everything. So that is the primary consumer of all ATP. Second is the cells which divide. And actually third comes the brain. But when you look at the systems, it's the brain and then everything else.
Just to share some trivia, the maximum amount of mitochondrial content is in skeletal muscles, followed by sperms. So,
Yes.
So, yeah. So any last comments, Dr. Shobha?
I think it was a nice presentation and we learned a lot. Sagar could tell us what you're going to cover in the next presentation so that maybe the audience can be prepared for it. That will be nice.
so ma'am, in the next session, we will be discussing more about the so-called mental health disorders, neurodegenerative and developmental disorders, and whether there is any role of bioenergetics impairment in these disorders or not.
Okay. So thank you. I
Thank you,
think
ma'am.
that's all.
Yeah. Thank you, Shobha, for chairing this session and asking so many questions and interacting with us. Thank you, Dr. Sagar. It was a nice presentation. And we eagerly look forward to the second one, which is going to be more about the disorders that we deal with day in and day out, and what more we can think of in terms of existing treatments and newer targets. Thank you everyone for spending your time with us today on the Brain on Wednesdays. And lastly, let me thank Dr. Raj Kumar sir. He is not here with us today. There are a lot of climate issues and heavy rains and power failures in Vadodara where he is located. And thank you, Alchem, for giving us this platform for these sessions. Thank you so much and good night. And in the next few days, we'll have the presentation available on YouTube. So you can have a look at it. Thank you. Thank you, everyone. Good night.
Thank you, sir. Thank you, ma'am.